Dear students, welcome to today's discussion, Basic Assumption and Principles of Experimental Research. So in our previous discussion, we have found that an experiment is a study in which a treatment, a procedure or program is intentionally introduced and a result or outcome is observed. For this, the experimenter has to prepare experimental design beforehand, which need to be based on some principles and assumptions. Let us see those basic assumptions and principles. So, the basic assumption of any experimental research is the law of single variable that was formulated by John Stuart Mill in 1873. He stated that if two situations are similar in every respect and one element is added to or subtracted from one but not the other, any difference that develops is the result of the operation that element added or subtracted. A simple example will be helpful to make it clear. Suppose two twin sisters in whom all other factors are similar, and sister A is given regular practice, whereas sister B is not. When the academic achievement on mathematics is taken, sister A performance is found to be higher. Here we may conclude that the single variable, the regular practice, may be the reason behind the difference of achievement between the two sisters. So Mill has put forward another 4 canons to explain all types of logical procedure required to explain control of events. Later on, it is observed that though the concept of single variable proved useful in some areas of physical science, in behavioral research, particularly when we have to deal with human behavior, this approach is not that much approachable or acceptable, as human events are usually the result of the interaction of many variables or many characteristics or abilities that cannot be explained in isolation, or we cannot explain or interpret it through single causes. At that time, R. Fisher came forward with the concept of equalizing of conditions through randomization and analysis of variance in pre-experimental phase itself. That helps the researcher in studying complex interaction effects through factorial design. So these are the basic assumptions of experimental research. The law of single variable along with another 4 canons and R. Fisher's factorial design of experimental research. A good experimental research design is based mainly on 5 conditions. These conditions are also known as the characteristics of experimental research also. 1 is control, random selection of samples, random assignments in groups, random assignments of treatments, and manipulation, then replication, observation and replication. Let us discuss this in elaboration one by one. So control. So control refers in experimental design 2 aspects. One is the formation or selection of samples in control group where the independent variable is not treated or the experimental treatment is not given, and another is control of the effects of extraneous variables that has the capacity to influence the cause and effect relationship between the independent and dependent variable. So variables or factors that the investigator wants to study are controlled by treating them as independent variable. For example, the investigator suppose want to compare program instruction in learning mathematics and wish to control the potential extraneous variable sex by including it as an independent variable. That means here sex is means the male and female or boys and girls students are matched or counterbalance, and thereby investigate the effect of sex on two teaching methods. On the other hand, the variable sex may also be controlled by eliminating sex, that means conducting the study with uniform characteristics only with the help of female students. So control is used to prevent outside factors from influencing the study outcome. So control in experimental and control group may be brought to matching, counterbalancing, and homogeneity by statistical tests like chi-square, ANOVA, ANCOVA, etc. Another condition or principle is randomization. So randomization provides the most effective method of eliminating systematic bias that minimizes the effect of extraneous variables, means the effect of those factors that are not treated in the experiment. So we all have an experience of playing Ludo or throwing dice, where we throw the dice randomly or arbitrarily, in which every face has the chance to appear. Similarly, in randomization, no matter who the participant is, she have an equal opportunity to be selected into all the groups or treatments in the group. Randomization helps to ensure homogeneity within the group. However, randomization is not a haphazard or arbitrary method of assigning subjects into groups. It helps in reducing the experimental error in the groups. Now, question sometimes may be arise, what is the experimental error? So, experimental error is an unintentional lack of validity or accuracy that may arise in an experiment due to design of an experiment system or tool, random errors, errors in selection of sample, human error, etc. So, randomization helps us in increasing the internal validity of the research. Randomization has different methods. We may take the help of coin, flipping coin. Another technique may be slip of paper, computer assisted random sequence, and example may be helpful in clearing the randomization process. Suppose to study the effectiveness of chalk and talk method and program instruction in teaching research methodology, and here the researcher has to randomize the extraneous variables by equalizing the or keeping in the homogeneity of the group. So in assigning subjects or sample in the experimental group and the control group, the experimenter has to use randomization technique. For that, he may give a serial number to all the subjects and employ tables of random numbers, or toss a coin as head and tail and may apply computer assisted table of numbers. Suppose the researcher has to select 50 students from 500. The researcher may put the serial numbers in a pot and draw the slip until it is 50. Similarly, researcher could assign teachers, classrooms, equipments, class periods on random basis. These are the three basic phases of randomization: random selection of subjects into groups, the control group and experimental group; random assignments of subjects into experimental treatment among different groups. So the researcher tries to randomly select the sample, else if it is not possible, assign the selected subjects into different experimental group and randomly assign a different experimental treatment into experimental groups. Another important characteristics is manipulation. So manipulation means something which is purposefully sensed by the researcher in the environment. This is the main difference between descriptive research and experimental research. In descriptive research, researchers simply study the status, observe what occurs naturally. But in experimental research, conditions are created artificially with some predetermined set of conditions which are treated or imposed on the subject of the treatment group to observe the variance or effect. Usually, an antecedent and independent variable consists of a single factor only, because an experimenter wishes to study the effect of a cause independent of all other influences. But with the help of statistical analysis or factorial design, multiple factors variance can also be calculated and analyzed. Next condition is observation. So observation is the process where experimenter observe the effect of the manipulation of the independent variable on dependent variable. Next is the replication. Replication means repeated application. Here the investigator use single identical procedure with different set of subjects in a different setting at different time. The repeated application of the treatments under investigation helps the experimenter to estimate the experimental error. It helps the experimenter to identify whether the observed difference or variation caused by the independent variable on the dependent variable is real or not. It also helps to reduce the experimental error. As I have said earlier, experimental error is an unintentional lack of validity or accuracy that may arise in an experiment due to design of an experiment, whichever is applicable. It may be pre-experimental, true experimental, or quasi-experimental. And true experimental design is the most acceptable design of experimental research. So replications helps in reducing the experimental error, and a satisfactory precision can be obtained by taking the number of replication So in the summary, it can be said that experimental research is based on valid estimation of experimental error, and for estimation of experimental error, the principle of randomization and replications are used, and to reduce experimental error, the principle of replication and control is used. So I hope you have an idea about the basic assumption and principles or characteristics of experimental research from today's discussion. If there is any confusion, you may drop a message, and I hope in the next class we shall discuss about the steps of experimental research.